is that bacteria can couple transcription and translation. And so in this particular diagram, you see a double-stranded DNA transcription bubble, and this is the messenger RNA here. And as the transcript elongates, ribosomes can actually sit on particular ribosome binding sites and initiate translation of different proteins. Obviously, this is made possible in prokaryotes because they don't have the separation that eukaryotes do between the nucleus and the cytoplasm. So eukaryotes obviously do their transcription in the nucleus, and then transport their messenger RNAs to the cytoplasm where it's translated. But again, there's no separation like that in prokaryotes. It facilitates rapid growth, and what's even cooler is that it also facilitates mechanisms of gene regulation, which I'll talk about. The next slide illustrates basic gene structure. This happens to be a monocistronic gene organization, meaning that there's a single gene, there's a single promoter, and a single transcription terminator. And this open reading frame here represents a single gene product. And I'll talk about polycistronic structure in a later slide. Over here you see the promoter and the main elements of the promoter, the -10 region and the -35. And again, in another slide, I'll show you specific -10 and -35 consensus sequences. These are where the RNA polymerase would look for. These regions here, the RNA polymerase would look for, and that would tell it where to initiate transcription. One other cool thing about this is down here is the transcription terminator region, and so that tells the RNA polymerase where to stop, essentially. And then here we look at the numbering. So +1 represents downstream of the start. So +1 is this base right here, of the transcription start codon. And then if you go to the left, which we would call upstream, the bases are numbered in the negative, so -10. So again, if we're going -1, -2, -3, so forth. So these are -10 and -35 and numbered because they're to the left of the start codon. And then anything downstream is +1, +2, and so forth. The -35 and -10 regions are roughly -35 and -10. They don't exactly precisely occur in those regions. And when you analyze a bunch of different promoters, you can kind of find homologous sequences, but they're not always precisely in those locations. So these are generalizations. The direction of transcription is indicated, proceeding downstream of the start. Here's the messenger RNA transcript here after transcription. And so you see a couple features of the messenger RNA transcript, the first of which is the untranslated region. Then the ribosome binding site, which the name suggests itself, where the ribosomes bind, the translation start codon, and the actual gene itself in blue, and then an untranslated trailer region. And so the so-called leader regions, the 5' and 3' untranslated leader regions, leader and trailer regions, are really important for a variety of things. The 5' is typically for gene regulation, which I'll talk about in a minute, specifically a mechanism called attenuation. It also is the home for basically for the ribosome binding site. If you consider this all untranslated, because this is not a region of messenger RNA that gets translated into protein. So technically all of this is the untranslated region. But included in that is the ribosome binding site. The 3' region here is typically for termination to facilitate translation stop. But it's also, where the poly A tail is added. In this region here, the poly A tail is added for, and one of the other cool things about it is that it's a source of small RNAs that can combine with a protein called HFQ to silence gene expression. So I had that little note here just in case you can get all that. But this region right here can basically be clipped off And then this RNA can actually associate with a protein called HFQ to literally silence certain genes from being expressed. And silencing RNA is something that we'll talk about later. The transcription start codon, ATG in the DNA anyway, and then the transcription stop codon is typically TAA. So if you're ever doing some bioinformatics work, often if I'm looking for genes, start and stop, I'll look for these particular codons. The trip operon attenuation model is what I was alluding to before when I talked about this untranslated leader region being essential for gene expression. So in this model of trip attenuation, there's, in addition to transcription initiation control, transcription continuation is also controlled. Attenuation is termination of a transcription within the leader region, and again, or the leader peptide, it's also called. But if you look at this particular diagram here, it's a little easier to see it all at once. In panel A here, no translation is happening. And let me back up and just say that the tryptophan operon is basically some genes that are essential for the biosynthesis of tryptophan. And so an organism that is in need of tryptophan would want to activate this operon. If the organism is flush with tryptophan, if tryptophan is present in the environment, say it's E. coli growing in TSA, for example, the organism is going to have plenty of tryptophan and would want to shut this operon down because it doesn't need to waste energy making biosynthesis enzymes. So in this particular panel here, they're just showing you no translation occurring. So looking at the anatomy of this a little bit, here's the tryptophan leader region. And then TRPE, as you see here, is a major gene encoding an enzyme called anthranilate synthase, as you see down here, which is a precursor to tryptophan. And so the other thing to notice here is that here's the RNA polymerase. And as the RNA polymerase is reading this untranslated region here and basically transcribing it, you have two different loops. There's a transcription pause loop. Region 1 here and region 2 can fold up on each other due to homology and make a loop. And then there's a terminator loop where region 3 and region 4 form. And when that happens, when the terminator loop forms, that kicks RNA polymerase off of the DNA and stops transcription. So looking at panel B, this is more of an actual scenario where translation is occurring in a low tryptophan environment. So again, you have to think about that as it's a low tryptophan environment, so the organism would want this operon expressed. And so the ribosome is moving along and it hits this TRP leader region here. And what's interesting is the TRP leader region has a lot of codons for tryptophan. And so that dictates the pace of the ribosome. If there's a lot of tryptophan around, the ribosome goes quickly. If there's not a lot of tryptophan around, the ribosome slows down. And that's why it says here ribosome stalls, because there's a lot of TRP codons and not enough tryptophan to keep the ribosome moving fast. And as a result, if this guy pauses just enough, then this other intermediate stem loop forms between regions 2 and 3 called the anti-terminator. And so the anti-terminator forms, preventing the terminator from forming. And as a result, the RNA polymerase can keep moving. Now, if we look at the opposite situation, translation is occurring under high tryptophan levels. So in this particular case, the ribosome is moving along and moving along quickly, right? Because there's high tryptophan levels. And as a result of its pace, it quickly covers up regions 1 and 2, which then allows the 3-4 loop, the terminator loop, to form. And that boots the RNA polymerase off of the DNA, and therefore transcription is terminated. This diagram just shows you the components of a typical prokaryotic RNA polymerase. And so the the specific functions of each of these components is here, and I'm not going to read that to you. You can read that for yourself, but I just want to run through it really quickly. There's two alpha subunits and a beta subunit, and these guys come together initially to form an alpha 2 beta, and then they combine with a beta prime and omega fragment to make the core enzyme. Now this is not a functional polymerase right here as an alpha 2 beta, beta prime, omega. It requires a sigma factor that you see here. And now it's the holoenzyme when all these components are together. And the holoenzyme can then find specific promoters and initiate transcription. Another cool thing about this is that sigma factors are modular. The RNA polymerases can bind to different sigma factors, which could then drag the polymerase to different promoters. And so there's a stationary phase sigma factor, for example, that can activate RNA polymerase to turn on genes that might be necessary in stationary phase. Or there's a log phase sigma factor that can initiate transcription of genes that are necessary for fast, rapid growth in log phase. And even more specific sigma factors for flagella synthesis or nitrogen utilization. So again, you can read through this just to get a sense for the different fragments and what they do. This diagram just illustrates one of those concepts that I talked about before, where I like to talk about antibiotics or things like that could be used to intervene in these processes. And so what you're seeing here is the RNA polymerase. Here's the sigma factor, and then different components of the RNA polymerase, the alpha and beta subunits, and here's a DNA template, and then the non-template. So this is the strand that's actually going to be used as a template to lay down RNA. And so all you're seeing here really is where the drug rifampicin or rifamycin, the two are pretty interchangeable, rifampicin or rifamycin. But here's where the drug would sit right in the pocket of this beta subunit and block RNA extension. And so RIFs are a drug that are often used to, for example, treat tuberculosis. So this slide is just illustrating transcription initiation, as I talked about before. And so again, you can see the coding strand and the template strand, and obviously the RNA transcript coming off of that. But I wanted you to hone in on the sequence of -35 and -10. So the -10 we typically call Tatabox for obvious reason. TA is a pretty common set of bases in the -10 sequences of a lot of different prokaryotes. The -35 consensus sequence is roughly something like this, TTGACA. Not perfect. I mean, you can line up the promoters of 100 bacteria, and you'll see roughly that sequence and roughly this sequence, but it's not perfect. The next slide is polycistronic mRNA. So I already introduced you to the concept of monocistronic, where you have a single promoter, an open reading frame that encodes a particular protein, and then a terminator. But here's a polycistronic messenger RNA where you have a promoter and an operator. The operator is where some kind of a repressor would bind in the instance where this needs to be shut off. And so after the operator, though, is a series of structural genes, and these things are all regulated by this common promoter. So an operon, by definition, then, is a single promoter that's regulating two or more contiguous genes. In this hypothetical example, it's 4 contiguous genes, but operons can be huge. They can have 10, 12 contiguous genes. So the polycistronic DNA is transcribed into a polycistronic messenger RNA. And so each of these regions would have then different ribosome binding sites on the same piece. And so that ribosomes could sit on these different ribosome binding sites and produce individual proteins. And again, this is all about efficiency. The microbe is being efficient and facilitating its rapid growth by coupling transcription and translation, and by having polycistronic messenger RNA. And so it looks something like this, where you have multiple ribosomes sitting on multiple ribosome binding sites, producing multiple proteins. I'm not going to go through this because there's enough text on here. You can read this and then go through take a look at what's happening, but it paraphrases. But I've already paraphrased what's happening here. You have a sigma factor, an RNA polymerase, an unwinding, and then obviously transcription. But make sure you read these and understand them. At the end of this, we end up with transcription termination, which occurs when core RNA polymerase dissociates from the template DNA. And the way in which that happens, there's a couple mechanisms of transcription termination, one of which is row independent versus row dependent, but we'll emphasize row termination. But in this particular case, a protein called NUS-A, which can bind to RNA, and essentially, and it can also bind to RNA polymerase, which in that complex can slow down the transcription, and as a result, you can get these stem loops to form that would also aid in pausing and then kicking the RNA polymerase off of the DNA. So NUS is really about controlling the rate at which the RNA polymerase is moving along and transcribing. And then the combination of NUS slowing things down and stem loop formation, typically in the terminator region, causes transcription termination. And then we add an extra layer to that by this protein called Rho. The Greek letter Rho, if you can see that here, looks for a specific hunk of DNA. I'm sorry, messenger RNA. Obviously, it's encoded in the DNA, but it's looking for a specific, because it's an RNA binding protein, looking for a specific site on the messenger RNA transcript called RUT. And I don't know if they did this on purpose by naming this RUT row. Old reference to Scooby-Doo. I don't know if that's way beyond your time, but Scooby-Doo used to say RUT row all the time. Anyway, and I have no idea if they intended that. But Rho binds to RUT and then slides up the RNA to ultimately catch up with the RNA polymerase to knock it off and to facilitate termination. But with NUS bound, NUS can actually regulate how quickly this moves. And so the combination of Rho chasing the RNA polymerase and NUS-A influencing the rate of RNA polymerase activity, there's a balancing act there. Again, NUS can slow down the polymerase so that Rho catches up faster and shuts things down quicker, or NUS can be a little more lenient and let RNA polymerase move faster and therefore facilitate more transcription. So you can think of NUS-A, and I put it down here, as an antagonist of Rho termination. It dictates rate, so it influences when Rho catches up and how much termination Rho can facilitate. Here we're just looking at ribosomes. I've already kind of alluded to that in one of the previous slides. Looking at this, and you see the different ribosomes, specifically here, where you can see the different subunits, the small subunit, the 30S, and the large subunit, the 50S ribosomal subunits. So that's illustrated here. And again, this doesn't make sense, right? That the 30S plus the 50S equals 70S, which is totally counterintuitive. But it's the same thing with eukaryotes. They have a large 60S subunit and a small 40S subunit, and counterintuitively, the two together are 80S. But it's all based on density separation in a density gradient. You don't need to know about that, but anyway, these S's are basically mass, units of mass. But the other component of translation is formylmethionine. This formylmethionine, here's the tRNA that specifically recognizes formylmethionine, and so tRNA fmet. This tRNA gets charged with a formylmethionine, and that is the typical initiator of translation. This particular tRNA will drag an fmet to the ribosome, and that's typically the first amino acid added to the polypeptide chain. So here's the methionine that makes up the first amino acid, and then there's a formyl group on there, kind of to label this as I'm the first amino acid in a prokaryotic polypeptide chain. So initiation is also reliant on Shine-Dalgarno sequence of the messenger RNA. And so specifically, the 16S ribosomal RNA and the 30S ribosomal subunit interact. And so that facilitates the binding of the ribosome, which again, you have to remember that ribosomes are made up of ribosomal RNA. And those ribosomal RNAs will have homology to the corresponding messenger RNA in the ribosome binding site. There's a variety of initiation factors as well, and these are required to form the initiation complex, and they're catalyzed by GTP.